All right, I'm going to talk about cell biology, and this is really as a foundation to talking about radiation biology. So the things I'm going to focus on with the cells biology are directly tied to how radiation affects the cells. Those parts of the cell which are not impacted by radiation, we're not going to touch on quite as much. So we want to appreciate some characteristics of a generalized cell, which is kind of what I was just showing you, like the organic cellular compounds, illustrates cell function and structure and composition, and then distinguish between different types of cellular reproduction. Those are all going to be the big ways that radiation can play a part in the life of the cell. So, what are the characteristics of cells? And sometimes biologists also just ask, what are the characteristics of life? How do we define what life looks like? And they move, grow, react, protect themselves, repair damage, regulate life processes, reproduce, and organize themselves. Radiation can impact cells at every single one of those levels, right? So we're going to look very specifically at every single one of those levels and talk about what radiation does that affects cells in all those different ways. So what I'm saying is, when we ask, what is life? That basic question of biology, we can also ask questions like, well, what isn't life? What actually goes against life? And radiation is one of those things when we look at a cellular level. So, what do cells require? And let me move this slide up a little bit because the aspect ratio is a little bit out. But they require food, which is generally some form of energy, oxygen to break down the food, and water to transport inorganic substances. Again, radiation impacts each part of that, and so we're gonna unpack that. And what does the body require from cells? Well, they help maintain homeostasis, normal function, despite outside influences like stress, exercise, injury, and disease. So these are the building blocks of life. Here is a slide depicting the overview of cell structure. Okay? So I'm going to very closely kind of move through cell structure and make sure that all of what that we're looking at makes sense. Okay? But I like this kind of illustration. I guess sometimes I like a map of things. I'm not necessarily a visual learner, but I do like maps if they help me orient myself. And there's one way to think about all the parts of cell structure that is tied to the appearance on the map of how all this stuff looks. So around the cell we have a membrane, and it is largely made out of lipids and proteins, and it's going to protect the cell and or control the passage of water and other materials in and out of the cell. And I'll review this too one more time. The protoplasm is a substance that exists outside the nucleus. So between the nucleus and the membrane there is protoplasm. And a lot of it, again, is water. And it's the place where all metabolic functions occur. So anabolism, which is like the assembly of proteins, catabolism, which is the breakdown of different things, packaging, waste elimination, all occur in the cytoplasm. It is kind of like the workplace of the cell. The endoplasmic reticulum, or the ER, is tubules and vesicles that facilitate communication and transfer of food. So one way to think about the ER is that it's like a highway system. The Golgi apparatus is, again, vesicles that extend from the nucleus to the membrane, and they are like a packaging center. They package large macromolecules like carbohydrates into glycoproteins. Okay, so one way I think about it is like FedEx, right? So endoplasmic reticulum is the highway, FedEx is the truck. Now, we need to have some kind of energy that's powering the truck to make it go down the highway. So where's the energy? Where's the powerhouse of the cell? The mitochondria. An interesting thing about the mitochondria, just a little factoid, is they're actually a separate species that integrated themselves into the environment of our cells at some point in the far, far distant past, right? So they are unique, and they have their own diseases and own life processes separate from the life process of the cell. They're a single-celled organism that migrated into our cells. Lysomes contain digestive enzymes, and one way to think about them would be like the garbage trucks of the cell. Okay? I think about lye, which is an extremely acidic substance. It can be used to burn things up. That's where the same word lysome comes from. And then ribosomes are used to synthesize amino acids and proteins. So they're like the little factories of the cell. So this just gives us an overview of where those things are located. And of course, the nucleus is going to be like the heart of the cell. And we'll talk more about it as we move forward. So here again is that cell structure stuff. I know I spoke kind of quickly. So I want us to really focus on these metaphors that we can use to think about this stuff. Cell membrane is going to be like the envelope that encases the cell. If you think about it, it's like city limits. It's the wall around the city, right? Cytoplasm is like the marketplace. It's where all the business is done, right? Endoplasmic reticulum is the highway system. It's the roads. It's the thing that connects everything within the city. The Golgi apparatus is like FedEx. It is the packaging system. It's what transports things around. That's the Golgi apparatus. Mitochondria are that power plant. Lysomes, we said, are like what? Garbage trucks, right? And particularly they contain digestive enzymes inside of them. That's going to be important. And then ribosomes are like the factories. They're where things are produced, right? Now, unfortunately, as we move along, this is about the place where the metaphor kind of goes off the rails, is with these two things here, right? Because they do things that no city would ever do, right? They do strange things. And so that's where our metaphor kind of falls apart. Centrosomes are located near the nucleus, and they contain centrioles, which are part of forming the mitotic spindle. Right? So centrosomes are a major part of cell division. They are what pulls the cell in two separate poles and creates a cellular equator that moves towards division, right? And the nucleus, I mean, if we're frank, the nucleus of cells is a very strange place, right? Maybe the best way that we could think about it would be it's like a library, right? I don't like thinking about it as the heart of the cell because it does a lot more than what a heart does. It stores information and potentially even has something like a memory. So it is filled with nucleoplasm, which contains DNA and proteins, as well as a nucleolus, right, which contains and manufactures and holds RNA and proteins. And then kind of spooled all throughout the entire nucleus is chromatin, which is the unspooled chromosome, right? It is all the DNA information that the cell requires to produce proteins, to respond to its environment, to send information, to eliminate waste. All of that's controlled by this code that's inside the nucleus. So the reason I want us to think about each one of these things is because each one of these places within our city is a place where a radiation event could occur, and it's going to then affect the city differently, right? So the largest part of the cell is going to be that cytoplasm, right? Because we've said that 80 to 85% of our body is a particular inorganic compound. Does anyone know what that inorganic compound is? Water, H2O, right? And that's the same is also true for cells. About 80 to 85% of every cell is water. And that's what fills up and is a large composite part of cytoplasm. So one of the biggest targets that we could have in our city is just the place where business is done, right? So when the radiation affects the cytoplasm, it's going to do unique things within the cytoplasm, right? Those we'll need to unpack quite a bit. but we might as well go ahead and talk now, even though we're still talking about biology, what radiation would do if it were to impact some of these organelles, right? If the radiation were to interrupt the highway system, what would we expect to happen? If a bomb was dropped in our city and it blew up some roads, how would we respond? I would hope we would fix the roads, right? That would be a start. The cell can fix the roads. That's not a problem, right? You just fix the roads. If a bomb went off in our city and it blew up the power plant, what would we do? We wouldn't have light. But just like the people in Houston or in Puerto Rico, we would have to figure out some alternate source of energy, right? So fortunately, the cell has numerous different mitochondria. If one of them goes down, another one can kick in and help, right? Now here's a weird one. What would happen if, in this world of the cell, the radiation hits a lysosome, which is full of digestive enzymes? Yeah, you would have a lot of trash, and in particular, this is like acidic trash, right? So, yeah, a lysosome can actually do what's called automatic suicide, right? Or it's called a suicide cell, where if a lysosome is ruptured, the cell, it spews acid into the cell, and the cell dissolves, right? Dissolves from the inside out. So lysosomes are a significant target. But can anyone take a guess on what's going to be the most significant target within our little cell city? The nucleus. And so the notion of a target theory comes from how radiation affects the nucleus. And we're going to look at that moving forward. So another way to think about, so what we just described is more of a biological explanation of how the cell is composed. We can also talk about the cell on a more chemical level. And just talk about the chemistry of the different parts of the cell. And this fits with kind of the model of radiation biology that we've put forward, that there's a physics to it, there's a chemistry to it, and there's a biology to it. Okay? So let's talk really carefully about what happens, or describe these chemical compositions, so that when we get to radiation biology, we can describe what would be happening. So within the cell, we've said that there is a cytoplasm or a protoplasm that exists between the nucleus and the cell membrane. And we've said it is primarily composed of water and some electrolytes or salts, as well as some proteins, right? It is just kind of the fluid in which the cell swims. The salts are going to help maintain proper cell performance. They are part of creating energy, and they conduct nerve impulses for nerve cells. Okay? The water within this protoplasm, again, is the medium in which metabolic activities occur. It is used as a solvent. And so you can think about if you got an ink stain or something on your shirt, we use water to kind of dissolve the ink stain or just dilute the ink stain. The exact same thing is happening within the protoplasm. And then it also works as a transport for different wastes, both within the cell and outside of the cell. And within the larger organism, water helps maintain body temperature, as well as it provides a cushioning for certain organs, like the liver in particular has a lot of water in it, and that's going to be part of how the liver protects itself. So again, those organic compounds that are within the cell, the number one compound that we think about for organic compounds is proteins. They're the building blocks for just about everything else. They are responsible for the growth and construction of new tissue. And they can be very simple things, which is what we call monomers, or they can be very complex things like polymers. So chromosomes and genes work together to create all the different types of proteins that are found in the body. There are some really, really important proteins, and those are repair enzymes, hormones, and antibodies. Okay? Repair enzymes initiate chemical reactions, and they can mend damaged molecules. So they feed right into that cell, that question of what is life. It is capable of repair. How does the cell do it? With repair enzymes, which are a protein. That's very important for us because healthy cells have repair enzymes. Things like cancer cells do not. So we can use radiation to kill cancer cells knowing they can't repair, and any normal tissue that's around it could potentially repair itself. Hormones are chemical secretions specifically from endocrine glands, and they're used to initiate activities throughout the body. Right? So growth, fight, flight, or freeze responses, accelerated heart rate, increased blood pressure, decreased blood pressure, all of those things are controlled by hormones. And then antibodies are protein molecules produced specifically by B lymphocytes, so white blood cells in the bone marrow, and it's initiated by T lymphocytes whenever they detect a foreign body or an antigen anywhere in the body. Each one of these is important, right? Because, for example, if radiation affects the affects a cell, it may be able to initiate a repair process, and we're going to look at that some more. If we irradiate particular areas where hormones are being produced, like the pituitary gland, or like the prostate gland, or other parts of the body where lots of protein or lots of hormones are produced, like the reproductive organs, that can affect the entire system of the body, right? One example I can say of that is whenever we were doing radiation therapy to the prostate, some of them became very angry, even belligerent, over the course of their treatment. Why is that? We have literally impacted the production of testosterone in their body, and they are responding to that hormonal change, right? Antibodies are a big one because these T cell and B cell lymphocytes are they're the most radiosensitive cells in the body, and so anytime we give radiation, we are potentially depressing the lymphocytes in the blood cells or in the bloodstream, and that could eventually cause the body to have a reduced immune response. Other organic compounds are carbohydrates and lipids, and they're largely responsible for energy and structure, right? So we're not as concerned with them. We just need to know what they are and what they do. But they're not necessarily significant to radiation biology. One exception to that is with carbohydrates. These are what we're exploiting with FDG and PET imaging. So we know that cells require a lot of carbohydrates to function. So these are things like sugars, like glucose. And so we tag that with a radiopharmaceutical, and we can look for those cells that are particularly avid for that sugar molecule and say that that's an area of increased metabolic activity, therefore it's an area of potential cancer. And the lipids, I think we've already discussed how lysomes can function. In terms of the inorganic compounds, I think we've already touched on them to some degree. But the big ones that we need to be familiar with are water. It's the number one. And then we've already discussed a little bit how within lysomes there are acids that could serve to dissolve the cell. So the inorganic compounds related to the body are acids, bases, salts, and water. The most important one for us is water. Now we should probably touch just a little bit on the chemical composition of DNA, and we'll come back to it too. But DNA is a sequence of amino acids that's used to produce proteins, it is the genetic code, right? And it's composed of two long sugar phosphate chains that are linked together by pairs of nitrogenous organic bases. And the way that I remember that linking is the mnemonic at G and C, like the health food store or whatever. So adenine's always going to bond with thymine, and guanine always binds with cytosine. And that's a nitrogenous base, so that's where I get the N from. RNA is structurally and chemically different from DNA. And it does a lot more functions than DNA. So DNA just houses the genetic code. RNA is able to translate the genetic code from the DNA into protein. So initially, when biologists were asking what is life, what communicates life from generation to generation, they thought that RNA was the target, that it was the storehouse of life because of all the functions that it performs. Basically, the DNA has a double strand. RNA is a single strand. It has a single sugar phosphate chain. And to make things confusing, RNA pairs uracil with adenine. So the mnemonic is like, G and C at that point, right? It doesn't really make any sense. But I always think about weightlifters going like, that's RNA, is G and C. So it has uracil paired with adenine. There is messenger RNA, which communicates from the nucleolus or the nucleus out into the cytoplasm, and then transfer RNA, which transfers that message from the messenger RNA into the ribosomes that produce the proteins. So messenger is the stuff that exits the nucleus out into the cytoplasm. Transfer is the stuff that transfers the message into a protein. Okay, well, how do we learn all this stuff? So it's helpful to have maybe a little bit of context around this. Hershey and Chase were early scientists who begged the question whether or not it was DNA or RNA that was responsible for the genetic code. And they designed an experiment, which, interestingly enough, used radioactive pharmaceuticals. And so the Hershey and Chase experiment looked like this. They had a virus, right? Which a virus communicates with RNA. And so this is one of the reasons why they thought that life, the message, the genetic code was carried in RNA, is because they could see it very clearly within viruses at that time. So what they did was they tagged the RNA with radioactive protein, and they did two different taggings. They did one that would have an affinity to RNA within the cell and one that would have an affinity with DNA within the cell. So they allowed the virus to infect the cell, and then as soon as the infection process, as soon as the RNA had been inserted into the cell, they froze the cell, stuck them in a blender, and blended them up. And they tried to see where exactly did the RNA go to, and it went to the DNA. So the way that the viruses were contaminating the cells was not through contamination of the RNA, it was through contamination of the DNA. And so that is one of the reasons that we know what we know about DNA. I want to stress this, is because of what we know about radiation, right? The theories and the ideas that we have about biology, we could not have arrived at without the discovery of radioisotopes and X-rays. Okay, let's talk a little bit about chromosomes and genes. This is a point of some confusion for students. But chromosomes are these tiny rod-shaped bodies. And normally there are 46 different chromosomes, and they're composed of 23 pairs within each somatic cell. Okay? Germ cells have only 23 chromosomes. That's half of 46. DNA can be broken down into different segments called genes. And a gene is basically just a complete code within the DNA. So it may be a sequence of nitrogenous bases that represents coding for something. And there's a bunch of different things it can code for. In terms of what we know about the genome, all the genetic material that we need is contained within these chromosomes. And so in 2001, scientists completed mapping the human genome, right? And they found roughly 2.93 billion, let's say, base pairs within a human genome. And these arrange into about 3,000 different genes, 30,000 different genes, let's just say, 30,000 different genes. And they're responsible for the production of about 90,000 different proteins, right? So those are some big numbers, but if you think about how complex an organism the body is, those numbers don't seem quite so big, right? What I'm saying is that there's only 46 of these chromosomes, and they code for all of these different billions of combinations. And those billions of combinations are used to produce about 90,000 proteins, and that is your body, right? That is your body from a coding point of view. So let's look a little bit more at DNA. Here is just a repeat of some of that information I gave you earlier. Deoxyribonucleic acid, it will carry that genetic code, and it is responsible for the production largely of proteins as well as the determination of what the cell's function is. Now, here's a weird question, right? I've said that every single cell in your body, with the exception of the germ cells, has 46 chromosomes in it. Every single cell has the exact same 46 chromosomes. In essence, every single cell in your body is a clone of every single other cell in your body. That's another way that you could say that. So let me ask you this. Why can't we grow like teeth in our eyes? The genetic code is the same for both. The cells in your teeth are the same. They have the same code as the cells that are in your eyes, right? The cells in your kidneys have the same information as the cells that are in your nose. So how is it that a kidney doesn't look anything like a nose and functions completely differently? Have you ever wondered that? It's a strange question. Part of what we're learning is it's not so much what's communicated as what's not communicated. So any cell in your body is a clone of every other cell in your body. The only thing that they differ in is what they don't communicate. There's epigenetic processes or processes that go on top of the genes that silence or allow certain parts of the genetic code to be expressed. And those epigenetic processes, they're called DNA methylation, are the reason why cells within your eyes don't all of a sudden say, I'm going to make a tooth cell now. Is they don't just communicate the genetic code, they also communicate epigenetic information. One way to think about it would be like when I make marks on these slides, right? So the slides are the genetic information, let's say. Whenever I make a mark like this, that is epigenetic. That is added to the slide. So something adds to the DNA information. I want to stress that because, frankly, we don't know what radiation does to those epigenetic processes. We can talk about what it does to the genetic processes, but we do not yet have any science that tells us what it's doing to epigenetic processes. So here again is a side-by-side slide talking about DNA versus RNA. One way to, if you know a little bit of Latin, if you're comfortable with that, deoxy means two oxygens, right, versus ribonucleic acid. Deoxyribose means that there are two sugar phosphate chains. When we just say ribonucleic acid, there's just one sugar phosphate chain. So that's the difference between deoxyribose and ribose. Double strand versus single strand. We said that the mnemonic for RNA becomes like GNC, right? Because of uracil. DNA is a very, very long chain of nucleotides. So if I were standing at the front entrance to this building, right, and I were to unspool a single chromosome, like a single strand of DNA, right, I would be able to get roughly to that park over there, Health Sciences Park, with just that single strand of DNA. And then it's just molecule by molecule stacked up. Deoxyribonucleic acid DNA carries a lot of information, right? And that's all it does. It just says that one function. It just carries information. The RNA has a bunch of different things that it does. It can be messenger RNA, it can be transfer RNA, or it can be rRNA or ribo RNA. It can actually be the protein factories. So here's again just a quick discussion of what those are. When the DNA translates, DNA never leaves the nucleus, first off. DNA never leaves the nucleus. It does not leave the nucleus. There's a brief period during cellular reproduction in which the nucleus itself dissolves, but the DNA has now packed itself up in chromosomes, and so it is essentially carrying the nucleus with it at that point. RNA is produced in the nucleolus, and it may be found in the nucleolus, it may be found in the nucleus, it may also be found out in the cytoplasm around the nucleus. So how it works is the DNA, when it recognizes, oh, I need to code for more endoplasmic reticulum, right? We need more of that stuff out there. It will transcribe that information to a messenger RNA. That messenger RNA, that mRNA, then exits the nucleus, goes out into the cytoplasm, is conducted through the smooth or the rough ER to a place where it can produce to a ribosome, to a factory. Within that factory, the ribosome unpacks or unzips that messenger RNA and uses a transfer RNA to transfer that data directly into protein structures, right? So each one of them perform different functions. They are very similar in their structure. So let's talk real quickly about cellular reproduction. There are two broad categories of cellular reproduction. I'm only going to focus on mitosis. For meiosis, which is the cellular reproduction for germ cells, all we need to know is that it results in daughter cells. The end product is going to be 4 daughter cells, each of which have 23 chromosomes. So it halves the number of chromosomes and reproduces 4 cells. That is germ cell reproduction. We call that meiosis. Mitosis is what we're going to focus on, and it produces 2 daughter cells, both of which have information identical to the parent cell, right? So they will, if the parent cell has 46 chromosomes, which is the norm, both the daughter cells will have the same 46 chromosomes, and they will also have the same epigenetic markup on that code. So here's a big slide kind of looking at that, but like I said, I'm going to zoom in just on mitosis. Interphase is the longest part of the cell cycle. Yeah, I just want to make sure that. And basically, it means the phase, interphase literally just means the part between mitosis, right? It's that interim phase. Within the interphase, though, there is G1, G sub 1, which is pre-DNA synthesis. It's a growth part of the cell cycle where it's growing, it's developing enough organelles to maintain all the cell functions during synthesis. The next phase it will go through is an S phase, which is the synthesis phase. It's a relatively long phase, and this is the time in which the chromosomes reproduce and split, forming two sister chromatids attached at a centromere. And then it will enter into G2, and this is post-DNA synthesis. It's still part of the interphase part. Now, it's important to note that during these parts of the cell cycle, the cell is its most radioresistant. Because of these growth and synthesis phases, the cell can slow down growth or slow down synthesis in order to initiate repair operations during interphase. So I'll say that again because it's important. Interphase is going to be the most radioresistant part of the cell's life because it has these two growth phases where it can initiate repair. The processes of mitosis are typically divided into 4 phases the way our textbook does, so we can ignore the proto-metaphase here. But there is prophase in which the nucleus changes to form the mitotic spindle. So on the outside of the nucleus you'll start to see those centromeres forming the mitotic spindle. It's visible under a microscope. During the metaphase, the fibers are going to connect to that mitotic spindle and they start to form an equator within the cell. So the cell itself will start to have a visible equator. If we were to stop the cellular reproduction, if we were to freeze time at this moment and look at it under the cell, we'd be able to clearly identify the different chromosome shapes under a microscope because they've packed themselves up very closely. During the anaphase, those different centromeres are going to migrate to opposite poles of the cell, and they're going to start to pull the chromatids in opposite directions. Right? And then telophase, you can think about this is the phase right before splitting. So we will see 2 separate nucleuses at this point. They start to form separate nuclear envelopes, and then the cell splits. A process called cytokinesis.